Look closely at a common fruit fly, and you will find a microscopic neural galaxy. Inside a space smaller than a poppy seed, an incredibly complex network is constantly processing light, scent, and motion. That tiny network matters to us because humans and fruit flies share roughly 60% of the same DNA. The basic biological machinery driving how we form memories, how we sleep, and how we process the world is actively running inside a brain no larger than a grain of salt. Medical researchers trying to treat neurodegenerative diseases like Alzheimer's and Parkinson's face a structural problem. To repair a broken system, you first need a complete wiring diagram, a map showing exactly how every single component connects to everything else. This scale comparison chart illustrates the size of that problem. A human brain contains roughly 86 billion individual neurons. Mapping every connection between them generates a volume of data that is currently impossible to process. Until recently, the most complex nervous system scientists had fully mapped belonged to a microscopic nematode worm, which has a total of just over 300 neurons. While that served as a useful baseline, a worm with 300 neurons doesn't navigate complex environments, interact socially, or form long-term memories. We needed an organism capable of higher cognition. The fruit fly bridges that gap. It is an organism small enough that modern computers can process its data, yet advanced enough to exhibit true intelligence. In October 2024, a global research group called the FlyWire Consortium published the first complete wiring diagram of an adult fruit fly brain. The resulting map, known as a connectome, details exactly how over 139,000 individual neurons link together through more than 50 million chemical connections, or synapses. To collect this data, researchers physically sliced a fly brain into 7,000 microscopic sections. They then captured 21 million images using an electron microscope. Artificial intelligence analyzed those images to identify individual cells. To correct the inevitable machine errors, an international community of scientists and volunteers spent years manually proofreading the exact boundaries of every neuron. All of this effort culminated in Codex, a publicly searchable database that functions like Google Earth for a living, thinking mind. We can now use this database to track a physical thought. When a shadow passes over a fruit fly in midair, the insect instantly alters its flight path to avoid a potential predator. This chart of the brain's vector wireframe shows how the reaction begins. A visual stimulus hits the fly's optic sensors and converts into a raw electrical signal. The signal travels straight into the subesophageal zone, or SEZ. The SEZ is a central hub at the base of the brain that acts as the primary router for incoming and outgoing information. Inside the SEZ, the network evaluates the incoming data from a shadow, calculates the threat, and dispatches a specific escape command through a highly structured series of downstream neurons. We can follow that command exactly as it reaches the motor neurons, which directly control the muscles in the fly's wings to execute the physical dodge maneuver. Researchers are no longer guessing how a brain processes a reaction. They can type a query into a database and watch the exact physical street address of a reflex light up. All of this precision in a tiny insect raises a practical question. How does tracking a fly's flight path help us understand human neurology? The underlying hardware and the physical rules governing how neural circuits transmit signals are highly consistent across different species. 3 out of every 4 known human genetic diseases have a direct parallel in the genetic code of the fruit fly. This biological overlap is why scientific research focused specifically on this insect has already resulted in 6 Nobel Prizes. By mapping the entire fly connectome, science now has a working baseline. We have a complete set of rules demonstrating exactly how a complex brain communicates with itself. The infrastructure built to create this map, the image segmentation algorithms, and the collaborative proofreading platforms, was designed to be scalable. Those exact digital tools are currently being deployed to map the brain of a mouse, which contains nearly 1,000 times more neurons than a fly. The ultimate goal is to continue scaling these methods until researchers possess the computational power to map a complete human brain. The human brain is moving from a state of biological inaccessibility to a permanent physical coordinate system. A moment of wonder. The common fly in your kitchen is carrying the architectural blueprint that will trace every neural pathway in the human head.